identified a class of endonucleases that are RNase-T2 enzymes, and the homologs in yeast, RNAY1, is only able to cleave tRNAs at the level of the anticodon region. What we found now with the same enzymes, but in Arabidopsis thaliana, is that these enzymes are able not only to cleave the tRNAs at the level of the anticodon, but also at the level of the D regions. So this is the first evidence of such enzymes able to generate the short TRF fragments, and it has not been shown so far.
Key to resistance in a plant is a class of receptors, intracellular receptors, called nod-like receptors or resistance genes, MBLRR proteins. And plants carry hundreds of these. Every plant species has a different set of hundreds of these. And so if you want to isolate new resistance genes, we have to explore the diversity of resistance gene sequences and then track it genetically and chase down which gene confers a specific new resistance that we want to identify and hopefully use in the future. So what we've done is a method that's based on sequence capture. It's complexity reduction sequencing, specifically focused on the intracellular receptors of plants, these so-called MBLRR or NLR genes. And we've been looking at wild relatives of potato as a source of new genes for resistance to a very important disease of potato called potato late blight. We want to bring in new genes for resistance, but we're constantly looking for new sources of resistance, and this resistance gene enrichment sequencing, or RENseq, this complexity reduction method, enables us to characterize the diversity of resistance genes with much greater precision than was ever possible before, especially combined when we combine the capture with specific biosciences long-read sequencing. It enables us to assemble the complete niblerome of any genotype that we choose to examine of specific classes of plants.
Our strategy actually differs from the strategy which was taken by the International Wheat Genome Sequencing Consortium. Specifically, we decided to learn how to sequence genomes of the size of the wheat genome by first sequencing diploid relative of wheat, Aegilops tauschii, and then use it as a reference sequence to assemble the wheat genome.
So what we've asked ourselves is, can we first of all take rapidly growing hardwoods like poplar, put into them novel biochemical pathways to produce specialty chemicals to begin with, which are much higher value than a petroleum replacement. So we've used molecules like phenylethanol, eugenol is another example. And what we're trying to see is, how can we work with the plant to take that photosynthetic carbon and increase the levels of these molecules to the point where we can not just compete with the natural production that occurs of, let's say, phenylethanol in the world, but how can we move more and more of that carbon from photosynthesis into these molecules and have them stored in such a way that we can remove them efficiently and also cost-wise, where we can be competitive not only with the natural sources but also with petrochemical processes. The material that's left behind is so-called lignocellulosic, and lignin is like the skeleton, it's like the bone material in plants. It's the most difficult to decay. So what we're trying to look at is how much of this carbon can we take out of the lignin pathway and put it into much more valuable materials while still maintaining the structural integrity of the plant.
So my research combines plant genomics with making fundamental discoveries in plant innate immunity. What I'm interested in is improving plant health. I work with wheat, I work with fungal pathogens, and I'm very interested in adopting new technologies to improve plant health. Currently I'm focusing on the interactions between wheat and yellow rust. So yellow rust is becoming a big problem in the UK and worldwide and in the developing countries. And what I'm looking for is durable solutions for discovering new sources of disease resistance to yellow rust. I am adopting sequencing technologies to rapidly map the sources of resistance.
Chloroplast genetic engineering has incredible opportunity in biomedical research and improving the medical treatment. For example, diabetes is a global epidemic now, It's all over the world, and insulin has been available for more than 50 years. Until today, it's not affordable for a large majority of the global population. The reason for this is the way it is produced and the way it is delivered. So we have used the Chloroplast technology to solve these problems. We cloned the human insulin gene, introduced into the Chloroplast genome, and we made that in lettuce Chloroplast. 70% of the leaf protein is insulin. So we dried the leaves, powdered them, made capsules, and orally delivered them, and showed that this is similar to injectable insulin. It lowered blood sugar levels.
I'm part of a research group that's interested in seed development and how ultimately oil-forming seeds might be engineered to produce more oil, ultimately with the goal of using them as biofuels. We used RNA-seq to reveal what kinds of changes there were in the transcriptome as the seed developed. And in the work that I presented at this meeting, we were focusing exclusively on splice variants because the majority of any plant genes that have been investigated, there is actually the production of different isoforms from the same gene over any time course or under any particular experimental condition. We were able to reveal some of these events and relate them to what is already known about the functions of those genes.
I chaired the Track 1 session at the Plant Genomics Congress, and it was a great cross-section of plant science. It included just about all aspects of genomics, including a form of omics that I'd never heard of called shovelomics. It was a bit tongue-in-cheek, but somebody was talking about root development and kind of coined the term shovelomics, so that was kind of fun. So it included lots of omics, it included the whole spectrum of science from research through breeding and production applications, and just really top-notch talks throughout the day.
We found out only recently that in the particular biosynthetic pathways that we are targeting, the terpenoid pathways, that we see that these pathways not only create diversity based on the huge amount of enzymes involved in this and their functional diversity, but also that plants utilize combinations of these enzymes to further enhance metabolic diversity. So what we see here is plants having a minimalistic evolutionary approach to using the same kind of enzyme, a single precursor, to produce tens of thousands of different compounds. And I think we can only learn from these master chemists to mimic these kind of approaches in developing combinatorial biosynthetic platforms for any kind of novel bioproduct that we can approach.